Hello, everyone. My name is ANFALHELU from Avalon University, last year medical student. I'll be describing septo-optic dysplasia in this presentation. Septo-optic dysplasia is also called Demore-Cyr syndrome, and it was first described in 1941 by Reeves, who redefined the syndrome as dysplasia or hypoplasia of the septum pellucidum, in addition to optic nerve abnormalities. Later on in 1970, Hoyt and colleagues redefined what SOD entails and found that the disease almost always had pituitary involvement. To your right, you see a child with septo-optic dysplasia, and as you can see, the first presenting symptom would be ocular abnormalities. So this child has aniridia, which is seen here, incomplete iris. Septo-optic dysplasia is theoretically hypothesized to be due to vascular constriction during the first trimester or 2 months of embryogenesis. The constriction of blood flow and nutrients would force the embryo to shunt the blood to larger areas of the brain, which would lead to minimal blood flow going to the smaller areas, such as the optic nerve, septum pellucidum, the pituitary gland, the eyes, and that would lead to abnormal growth of these structures. Septo-optic dysplasia is considered a rare disease. It's estimated to present in 1 in every 10,000 births, or 8.3 per 100,000 live births. Its prevalence is equal in both genders. Studies show that it was found to be associated with young maternal age, maternal bleeding during the first trimester, and drug or alcohol use. It's highly variable, ranging from very mild to very severe, depending on how dysplastic the structures are. Although genetic mutations among family members with septo-optic dysplasia have been found, 99% of the time, it's an isolated sporadic finding. So in order for us to conclude a diagnosis of septo-optic dysplasia, there has to be a triad of midline defect, pituitary defect, and optic nerve hypoplasia. The triad can be detected through a coronal or axial T1 or T2 MRI, a fundoscopic exam, and repeat hormonal levels should be tested to determine just how much the pituitary gland is affected. Just like all rare diseases, a geneticist would need to do a full evaluation, which includes a personal and family history. And common questions asked should be about whether there is consanguinity in the family, hearing abnormalities, developmental delay, anosmia, other congenital anomalies such as cardiac esophageal atresia, micropenis seizures, stereotypical behaviors, sleep disorders, excessive appetite, temperature instability, and just basically symptoms suggestive of any hormonal deficiency, which could include fatigue, constipation, brady or tachycardia, numbness or tingling, amongst others. The most severe type of septo-optic dysplasia is referred to as septo-optic dysplasia plus. This type has the triad in addition to cortical malformations, and I'll be presenting a case about it at the end of this presentation. Since I'm presenting a syndrome, I found it best to split the 3 abnormalities in detail and explain them. So the septum pellucidum is a thin triangular sheet that lives under the corpus callosum and functions to separate the anterior horns of the right and left ventricle. And this symptom is associated with many conditions such as corpus callosum agenesis, holoprosencephaly, alobar, semilobar, and middle interhemispheric variant, schizencephaly, Avid syndrome, which consists of a asymmetric ventriculomegaly with interhemispheric disc combined with genesis of the corpus callosum. An isolated finding of a missing septum accounts for 2 to 3 per 100,000 cases in the general public. Some cases of hypoplasia are symptomatic, although studies conducted on prisoners and between prisoners and people randomly found that the cavum septum to be somewhat linked to psychopathies and disruptive behavior. So they differentiated between the prisoners and someone randomly in public and found that majority of the prisoners that had psychopathic abnormal behaviors had a hypoplastic septum. This is a FLAIR MRI on a normal brain, which is the normal brain is image B, and you can see the brain with the missing septum lucidum. You can appreciate that there is boxing of the frontal horns, and they are pointing of boxing of the ventricles, and the frontal horns are pointing inferiorly. You can also appreciate that the posterior pituitary is also descended compared to normal here. Ventricles are normally just triangular shaped. And the posterior pituitary to be descended is actually a common finding because in a structure, if you have a proximal structure that is hypoplastic, the distal structures would, logically speaking, be affected as well and displaced. The next abnormality we'll be talking about is optic nerve hypoplasia. Optic nerve hypoplasia is usually the first presenting sign of possible septo-optic dysplasia. It's due to the associated eye issues that result, which entails nystagmus, which is abnormal movement of the eye, coloboma, which is when there is missing tissue in parts of the eye, like pupil, lens, or retina, and iridia is missing iris, microphthalmia, microcornea, and among a series of other eye abnormalities. Optic nerve hypoplasia can be unilateral or bilateral, which actually determines how severe the visual impairment is. An interesting concept to know is that a hypoplastic anterior pituitary alongside an absent infundibulum is usually associated with bilateral optic nerve hypoplasia. The optic nerve alongside posterior eye structures can be visualized with a fundoscope done by an ophthalmologist. And if you look to your right, and you can see the fundoscopic image, which shows a small pale optic nerve. It also shows peripapillary atrophy, which is referred to as the double ring sign, and this is diagnostic for optic nerve hypoplasia. This is an image, a T1 weighted image, coronal and axial MRI. And here you can see that the optic nerve is hypoplastic compared to this one here as well. And also the boxing of the ventricles and the inferior pointing frontal horns. The last of the triad is pituitary gland hypoplasia. So the pituitary gland, as I like to call it, is the powerhouse of the body, and it functions to maintain homeostasis by sending signals to various organs. So imagine if the powerhouse is not working properly, and that is what we would call an hypopituitarism. The most common deficiency is of the growth hormone, if not all hormones are affected. So growth hormone can lead to deficiency, can lead to hypoglycemia, tooth development, delay, micropenis, slow growth, delay puberty. A patient with severe hypoplasia presenting with hypopituitarism will additionally have diabetes insipidus, hypothyroid, adrenal crisis, temperature and blood pressure instability, arrhythmias, and amenorrhea. So the image on the bottom right shows the different causes and effects of pituitary gland-induced function. Infants often present with a series of electrolyte disturbances as the initial feature if it's very severe, like panhypopituitarism. And they could have micropenis or microphalus with prolonged jaundice in addition to that. So this is a T1 weighted sagittal MRI image of the pituitary stalk. Image A shows a normal corpus callosum up here, optic chiasm, and this is the pituitary stalk, the anterior pituitary, and the posterior pituitary. Image B shows a patient with a homozygous HESX1 mutation, which is actually the most severe genetic mutation, and we'll go on to discuss that later. You can clearly visualize the severely hypoplastic corpus callosum, the optic chiasm, and the anterior pituitary in an empty cella. Note here also the ectopic posterior pituitary. And there's no pituitary stalk here. Image C is of a sibling with the same mutation, and here you can see the splenium of the corpus callosum is more hypoplastic, and there's a shallow cella, and the posterior pituitary is partially descended. So even though they have the same mutation, they were affected differently. And this is why it's highly variable. Several etiologies have been postulated to account for sporadic occurrence, such as viral infections, environmental teratogens, and vascular or degenerative damage. However, the precise etiology of the condition still remains unknown. Most likely it's multifactorial. It could be a genetic cause as well as environmental causes combined. Familial cases are rare but implicate a genetic defect underlying developmental mechanisms and can be recessive or dominant. And they're known to involve these three genes, SX1, SOX3, and SOX2. In the past, it was thought that the PAX6 gene was involved in septo-dysplasia, but there was no variants found at all, and they thought it was involved because PAX6 gene is a transcriptional regulator and a master control of eye development, but nothing has been set in print yet. SX1 is the first gene we're going to talk about, and it's a member of the paired-like class of homeobox genes and functions as a transcriptional repressor with repression domains within the N-terminal region and DNA binding homeodomain. It's one of the earliest markers of murine pituitary development expressed during gastrulation in a region known to form the forebrain. So SX1 expression is restricted to ventral diencephalon by day 9 and also on the thickened layer of the oral ectoderm, giving rise to Rathke's pouch, which is the primordium of the anterior pituitary. It's continuous development till day 12, and it should be undetected by day 13 and a half. Homozygous disruption of SX1 was found in mice, and it was associated with a phenotype that is very closely linked to septo-dysplasia. So the abnormalities are fully penetrant, although very variable. Mutations range from complete absence of midline structures, such as the pituitary, corpus callosum, to partial formation. Complete absence of these structures involved a more severe phenotype causing microcephaly, craniofacial dysplasia, and there is no sign of the telencephalic vesicle or infundibulum, and absent olfactory placodes. Heterozygous mutations, on the other hand, yield a mild phenotype, leading to most commonly growth hormone deficiency with or without undescended posterior pituitary, although optic nerve hypoplasia and midline structure abnormalities may also be associated. SX1 mutations are rarely to be associated, if at all. According to Kelberman and Ditani, they conducted a study that screened 800 patients with septo-dysplasia and hypopituitaries and found mutations in less than 1% of that. So sequence variants have been identified, including a conserved base in a cis-regulating region upstream of SX1, but it is still up to debate. The fact that SX1 mutations are rare means mutations in other known or unknown genes contributed to this syndrome. Unlikely factors from the environment also have an effect. The combination of that. SOX2 is the second gene we'll talk about. And SOX2 is a transcription factor, which is important in maintaining pluripotency of embryonic stem cells. It's of the subfamily just as SOX3 and SOX1 are. And it's located in a gene desert. A gene desert is a region of the genome which is devoid of protein coding genes, and it was thought once to have only junk DNA. Initial expression is detected at the morula stage and then in the inner cell mass of the blastocyte. After gastrulation, it's restricted to the presumptive neuroectoderm, expressed throughout the brain, the CNS, the sensory placodes, branchial arches, gut endoderm, and the trachea and esophagus. The homozygous loss of SOX2 would lead to peri-implantation lethality. Heterozygous appear normal but show reduction in size and male fertility. Studies show that a reduction of SOX2 levels below 40% resulted in anophthalmia. It also has a role in the regulation of correcting gonadotropin production, which explains the infertility. Studies indicate that there's a role of SOX2 in urine pituitary development, and heterozygous mutations manifested a variable hypopituitary phenotype, leading to reduced levels of growth hormone, LH, ACTH, and TSH. SOX2 encodes a single exon encoding 317 amino acid proteins containing an N-terminal domain of unknown function, a DNA binding HMG domain, and C-terminal transcription factor activator domain. So here is an image of the gene, and there have been 12 heterozygous de novo mutations reported, according to research by Kelberman and Ditani. In 14 patients that had anophthalmia or severe microophthalmia, in addition to developmental delay, learning difficulties, esophageal atresia, and genital dysmorphia. These de novo mutations included 5 nonsense, 4 frame shift, 1 deletion, and 2 missense. There was also found a loss of function in the C60 region right here, C387 right here, Y160, and C.47. So it was complete loss of function. So that would be the most severe type. SOX3 is a member of the SOX family transcription factor, which encodes an SRY-related HMG box. It's initially identified based on homology to the conserved binding motif of high mobility group HMG class that is present on the mammalian sex-determining gene, which is the SRY. It's encoded by a single exon, producing a transcript of 1.3 KB, which maps to chromosome XQ27. And I note this here because the specific gene would lead to not just an autosomal recessive or dominant, it would lead to an X-linked mutation in the family. SOX3 protein has a short 66 amino acid and terminal domain of unknown function, 79 amino acid DNA binding, HMG domain, and a longer C-terminal domain containing 4 polyalanine stretches shown to be involved in transcriptional activation. The expression of SOX3 is similar to SOX2 in the CNS, brain, spinal cord. There's high levels of expression also noted in the ventral diencephalon, including the infundibulum and the presumptive hypothalamus. Targeted disruption leads to variable complex phenotype, including craniofacial abnormalities, midline CNS defects, and a reduction in size and fertility. Heterozygous females are mosaic due to the X inactivation and may appear normal, or sometimes they have mild craniofacial phenotype, dysplastic phenotype. SOX3 it's not expressed in Rathke's pouch, unlike the HESX1. It's expressed in high levels in the ventral diencephalon, including the infundibulum, which provides necessary inductive signals for the anterior pituitary formation. So even if it's not directly involved in pituitary development, it's indirectly involved one way or another. Tandem duplications involving chromosome X were identified in several pedigrees with mental retardation and hypopituitarism. Studies show that both duplications of XQ27 expressed SOX3 and loss of function polyalanine expansion. And they're all associated with a similar phenotype due to the infundibular hypoplasia, which is critical for normal development of the diencephalon and infundibulum of the anterior pituitary. So you can see here there's a wide variability in the severity of the clinical features found and their association with other diagnoses. There's no clear pattern. The image shows that the main reported clinical findings are hypopituitarism with growth hormone deficiency, which accounts for 62 to 80%. Next is the vision impairment, which shows it to be severe at 23% significance. So 23% found significant visual impairment. And that is associated with developmental delay. There's also, it's also more common with bilateral optic nerve hypoplasia. So unilateral will have mild vision impairment, bilateral would have more severe, in addition to all of these right here. There was also cerebral palsy, seizures are the most common neurological things that are found here. Pituitary hormone insufficiencies may evolve over time, necessitating lifelong medical follow-up, meaning the patient born as an infant would not appear to be deprived of hormonal levels, but as time progresses, and usually it's after puberty or before puberty is when the patient will start to show any signs of deficiencies. So this is why it's important for a team of medical specialists to follow up throughout the life. So our first case would be about a 1-week-old newborn that presented to the NICU, and he had prolonged jaundice refractory to phototherapy apparatus. On presentation, he was a newborn male, born 35 weeks to a young mother, and he showed bilateral undescended testes, micropelma, nystagmus, microcornea, cataracts, aniridia, optic nerve hypoplasia, and colon. So all the eye abnormalities seen here, that's because that's bilateral optic nerve hypoplasia in addition to that. So he has the most severe phenotype for visual impairment. A cranial MRI revealed absence of the septum pellucidum and slight hypoplasia of the pituitary gland and a normal corpus callosum. Genetic testing was done for PAX6 and SOX gene, which were found inconclusive. So even though there is no link to the PAX6 gene yet, it's still tested because there might be. This is such a rare disorder. We don't know how environmental factors can affect each gene and lead to something different. The second case is about a 11.5-year-old Caucasian Southeast European female with an earlier established diagnosis of growth hormone deficiency, diabetes insipidus, seizures, mental retardation, optic nerve atrophy, and rhitosis. The girl has a short stature and low weight for her age, and she has bilateral optic nerve hypoplasia. So she has poor vision, nystagmus, and right eye oculomotor palsy. EEG revealed also epileptic changes. MRI showed an empty cella syndrome. There was partial hypoplasia of the corpus callosum, cavum of the septum pellucidum, and diffuse polymicrogyria of the left temporal lobe. So here the difference is this is a septo-optic dysplasia plus syndrome because there's cortical involvement. This is a coronal T2 weighted image of the girl. And here the MRI shows absence of the septum pellucidum, so there's a flat roof. And you can see also here the point down appearance to the inferior parts of the frontal horn. And there's loss of normal gyral architecture also of the left temporal lobe. So right here, you can see loss of architecture. There's a thickened cortex, and there's indistinct cortical medullary interface, which is what is referred to as polymicrogyria. And also there's an enlarged sylvian fissure. So the T2 weighted image does not really show midline structures as well. So that's when we appreciate the boxing of the ventricles and the inferior pointing of the frontal horns shown here clearly. And that is characteristic of septum pellucidum dysplasia. So in conclusion, we have talked about how septal dysplasia is a rare syndrome that is most often sporadic. Genetic mutation involves the HESX1 gene, SOX2, and SOX3. We also discussed how septo-optic dysplasia may or may not be diagnosed prenatally, and how diagnosis requires the triad of optic nerve hypoplasia, septum pellucidum dysplasia, and pituitary hypoplasia to be concluded. Detail was also given about its a highly variable phenotype and genetic involvement. Diagnosis of septal dysplasia entails a need for several specialists and a team that collaborates together for the patient and regular follow-ups every 3 to 6 months. Septal dysplasia plus was described as an additional finding of cortical malformation in addition to the triad. So recommendations include a more thorough prenatal screening during the 20-week anatomy scan, giving focus on the smaller midline structures. Additionally, further studies should be considered in order to evaluate the possible role that septum pellucidum has on human behavior. These are references, and this concludes the end of the presentation. Thank you.
Still recording. Let's see here.